Each of these green crescents is a single living cell. They're called Closterium. From a distance, they seem perfectly still, quietly suspended in water, doing nothing at all. But take a closer look inside, and you'll find a chaos of movement. This is cytoplasmic streaming, a transport system that has been flowing through these organisms for hundreds of millions of years. Without it, these giant cells could not survive. Closterium is a single-celled green algae, part of a group called desmids, found in freshwater ponds and lakes around the world. These crescent-shaped cells are giants in the microscopic world, often reaching 200 micrometers or more in length. Let's take a quick tour. At each end are large terminal vacuoles, fluid-filled compartments that contain the crystals we'll see in a moment. In the center is a narrow bridge called the isthmus, and tucked inside it is the cell's nucleus. Along the sides are two ribbon-like chloroplasts, the photosynthetic engines that power the cell. At this enormous size, Closterium faces a serious challenge: how to transport materials efficiently across such a long distance. That's what makes them ideal for studying cytoplasmic streaming. Usually when we think of movement in Closterium, we often notice this first, the dancing crystals at each tip. They're made of barium sulfate, and they jiggle constantly due to Brownian motion, random movement caused by collisions with surrounding water molecules. It's mesmerizing to watch, and while this random motion isn't directly helping with transport, some researchers believe these crystals may play a role in sensing gravity, settling toward one side like microscopic statoliths. But the truly vital movement is happening elsewhere. To understand why Closterium needs cytoplasmic streaming, we first need to grasp the scale problem. These cells can grow over 200 micrometers long, absolutely massive by cellular standards. Picture this. If a typical human cell were the size of a marble, Closterium would stretch as long as a baseball bat. In small cells, molecules move by diffusion, randomly bouncing through the fluid until they reach their target. You can see this at work inside a tiny paratrich ciliate. There's no active circulation here, just particles drifting gently from place to place. At this scale, diffusion is enough. But diffusion has a fatal flaw. It slows dramatically as distance increases. For a molecule to drift from one end of this cell to the other could take months. By then, the cell would be dead. At this size, passive diffusion just isn't fast enough to keep the cell alive. What Closterium needs is something better, something active. A system that can move materials purposefully, from where they're made to where they're needed. And that's exactly what cytoplasmic streaming provides. Here it is, cytoplasmic streaming in action. Watch closely. This isn't random jittering like the crystals at the tips. This is directional, organized transport. The cytoplasm flows in coordinated patterns within each half of the cell, carrying organelles, proteins, and nutrients along defined routes. You can even see bidirectional flow, streams moving in opposite directions like traffic lanes on a microscopic highway. And look here, movement crossing the narrow bridge at the center, threading through the isthmus where the nucleus sits. Those ribbon-like chloroplasts, they're not just floating freely. They're arranged along the streaming paths, riding the current like passengers on a moving walkway. This system moves materials at several micrometers per second. Scaled up to our size, that's like walking at a brisk pace. For a single cell, this is lightning fast. But how does this actually work? What creates these organized currents flowing through the cell? The answer lies in molecular motors, tiny protein machines that walk along internal tracks, pulling their cargo through the cell. The tracks are made of actin filaments, protein cables that crisscross the cell like an invisible highway system. The engines are myosin motors, especially myosin 11, the fastest motor protein known to science. These motors run on ATP, the cell's energy currency, using it to fuel each step. Watch this remarkable footage from Protist lab films. You can actually see organelles being pulled along these tracks, each one hitching a ride on a molecular motor. As they move, they push and stir the surrounding cytoplasm, creating the streaming currents that carry everything else along. It's like a microscopic conveyor belt system powered by millions of tiny engines, all working in sync. This molecular machinery has been fine-tuned over hundreds of millions of years, transforming what could be cellular chaos into elegant, organized transport. Now for something remarkable, an experiment I ran to answer a simple question. If streaming moves organelles through the cell, what exactly is being moved? To find out, I used a fluorescent dye called Mitotracker Red. It tags mitochondria, the cell's power plants, causing them to glow under special lighting. These glowing structures are mitochondria. They produce ATP, the energy currency that powers almost every active process inside a cell, including the molecular motors we saw earlier. Without mitochondria, the motors would stall, the traffic would stop, the system would grind to a halt. Watch now as we shift from normal light to fluorescence. The transformation reveals something beautiful. Some of the streaming material begins to glow, showing us that mitochondria are being carried along by the flow. But not all of it lights up. Only the active mitochondria fluoresce. There may be others in the mix, along with proteins, vesicles, and other cargo, all swept along in this microscopic river. What we're watching is a living supply chain, delivering energy and materials exactly where the cell needs them. So why does any of this matter? Because without cytoplasmic streaming, these cells couldn't survive. It solves three problems in one elegant stroke. First, distribution. Sugars made in the chloroplast must travel quickly and efficiently to the farthest reaches of the cell. Second, cleanup. Waste products need to be swept away before they build up and cause harm. Third, logistics. Organelles like mitochondria must be moved and positioned where they're needed most. Streaming handles all of it, continuously, silently, and with astonishing coordination. And Closterium isn't alone. This process plays out across the plant kingdom, from the smallest algae to the largest leaf cells in your backyard. It's a quiet revolution, one that helped cells grow larger, faster, and more complex than ever before. In a world where physics imposes limits, cytoplasmic streaming rewrites the rules. From the outside, Closterium appears still. Inside, it's anything but. Molecular motors walk along actin tracks. Mitochondria move with the current. Organelles are positioned, nutrients delivered, waste removed. All of it made possible by cytoplasmic streaming, an ancient and highly efficient transport system. It's not unique to Closterium. The same mechanism operates across the plant kingdom, solving the same problems in the same way. What you're seeing isn't just motion. It's a quiet solution to the challenges of being microscopically large, while still being just one cell. Thanks for joining us on today's look inside the microcosmos. We hope you'll consider subscribing to our channel so you won't miss our next adventure.